Okay, hello. So today I'm going to discuss about some question from biotechnology and its application. So next question is here. Question number 20. Which of the endonuclease is mostly used in genetic engineering? So the correct answer will be option B. Type 2 endonuclease is mostly used in genetic material. Next question. The function of restriction enzyme is option A cleavage of fat, option B cleavage of DNA, option C cleavage of starch, cleavage of protein. So the correct answer is option B cleavage of DNA. Next question. Manipulation of DNA in genetic engineering become possible due to the discovery of option A primase, option C transcriptase, option D DNA ligase, option C restriction enzyme. So the correct answer is option D. Next question. A restriction endonuclease is used in option A tissue culture, genetic engineering, option C cell fractionation, and D regeneration of the tissue. So genetic engineering will be the correct answer for this question. Genetic engineering. Genetic engineering is the correct answer. Next question. The advancement in genetic engineering has been possible due to oncogenes, exonuclease, transposons, and endonuclease. So the correct answer is endonuclease. So next question. Pentadiplandra brasiliana produces a protein called brasin, which is about 2000 times as sweet as sugar. This plant belongs to Canada, West Africa, Australia, and South America. So the correct answer is option C, West Africa. Next question. Which is used in molecular genetic engineering? So option A carrot, option C tobacco, option B tomato, option D Arabidopsis. So the correct answer will be Arabidopsis. So the correct answer is Arabidopsis. Which next question? Which one of the following technique is used for early molecular diagnosis? Polymerase chain reaction, recombinant DNA technology, polyacrylamide gel electrophoresis, enzyme linked immunosorbent assay. So the correct answer is option B recombinant DNA technology will be the correct answer. So okay, next question. Which one of the following technique made it possible to genetically engineer living organism? Hybridization, X-ray diffraction, heavier isotopes labeling, and recombinant DNA technology will be the correct answer. So genetically engineering of living organism is done through recombinant DNA technique or technology. Next question. So gene recombinant technology is used for A. Vectorless gene transfer into the target cell, direct transfer of DNA protein complex, Option C, vector-based gene transfer into target cell. Liposomes-based direct gene transfer into the target cell. So the correct answer is option C will be the correct answer for this question. Next question. So let's go to the next question of this. So let's question number. When a typical restriction enzyme cuts DNA molecule, the cuts are uneven so that the DNA fragments have single-stranded ends. These ends are useful in recombinant DNA work because they serve as the option A, serve as starting points for DNA replication. No, this is not correct. Only single-stranded DNA segments can code for core protein. This option also not correct. They enable researchers to use the fragments as molecular probes. And the option D, the fragments will bond to other fragments with complementary ends. So the correct answer will be option D. This also not correct. So the correct answer is option D for this question. Next question. Question number 37. In recombinant DNA experiment, dash is used to cut piece of DNA and dash joins these fragments to form recombinant DNA. So here option D will be the correct answer. Restriction enzyme is used. Restriction enzyme is used to DNA and DNA ligase is used to form the recombinant DNA. So for this question, the D option will be the correct answer. Next question. Question number 37. Restriction endonuclease are utilized in genetic engineering as a molecular build up at nucleotides, molecular degradation to DNA breakup, molecular scalpel for cutting DNA at specific sites, and option D, molecular cement for combining DNA bits into long chains. So the correct answer for this question will be option C, the molecular scalpels for cutting DNA at specific sites. So the function of a restriction nuclease. Okay, next question. Restriction enzymes cut or join DNA fragments. This option not correct. Are required in vectorless direct gene transfer. This also not correct. Are endonucleases which cleave DNA at a specific site. This answer is the correct answer. So next question. Let's go to the next question. Restriction endonuclease, an enzyme used in genetic engineering, is employed for cutting double DNA strand, cutting single DNA strand, copying DNA strands, and join strands of the DNA probing taxons. So the correct answer is option A, the cutting double stranded DNA molecule at a specific site of DNA molecule. So next question. So this question have comes AIMS exam, all Indian. So the next question. The restriction enzymes are present in several microorganisms cut foreign DNA at a specific site and destroy them. The enzymes do not destroy the cellular DNA because the cellular DNA does not have the specific site. No, this option is not correct. The susceptible specific sites are masked by protein. This also not correct. A restriction enzyme susceptible sites are modified by cellular enzymes. So this option is the correct option for this answer. The restriction enzymes and DNA occupy different compartments, so this option is not correct. Let's move to the next question. So the molecular seizure, which cut or seizure or seizure, which cut DNA at a specific site. So the correct answer will be the restriction endonuclease. So okay, next question. Which of the following cut DNA at a specific site? So the correct answer is restriction enzyme. Let's go to the next question. In nature, function of restriction enzyme is to cut plasmids, destroy fast DNA, splice DNA in a cell, destroy foreign DNA in animal cells. So the correct answer is option B, destroy the fudge DNA. So next question. Bacteria protect themselves from virus by fragmenting viral DNA upon entry with ligase, DNA ligase, exonuclease, gyrase, endonuclease. So the correct answer is endonuclease. Restriction endonucleases are most widely used in recombinant DNA technology. They are obtained from restriction again I'm reading the question again. Restriction endonucleases are most widely used in recombinant DNA technology. They are obtained from plasmid, bacteriophage, bacterial cells, and all prokaryotic cells. So the correct answer is here option B, bacterial cells. Next question. Let's move to the next question here. Then the question number 27. Question number 27. So which enzyme acts as a biological seizure in genetic engineer? So again the question answer is restriction endonuclease. So move to the next question. Restriction enzymes was discovered by, the correct answer will be the option C. This option is for this question. The question number 25. Which one of the following hydrolyzes internal phosphodiester bond in polynucleotide chain? Ligase, exonuclease, protease, and endonuclease. So the endonuclease will be the correct answer. Next question. The enzyme used to cut DNA molecules is DNA ligase, DNA polymerase, 2-galactosidase, RNA polymerase, and again the question is restriction endonuclease. So these are the questions. The enzymes commonly used in genetic engineering are restriction endonuclease and polymerase, restriction endonuclease and ligase, endonuclease and ligase, and polymerase. So the restriction enzymes and ligase is used in recombinant DNA technology or genetic engineering. So next question. Which of the following pair is correctly matched? So central dogma is correctly matched. Central dogma codon. Okazuki fragment, it's not correctly matched. DNA RNA polymerase, this also not correctly matched. Central dogma, it's also not. So the correct answer is restriction enzyme and genetic engineering is correctly matched for this question. So let's go to the question number. Here there is a restriction endonuclease called EcoRI. So what does co part in stand for? So the correct answer will be A, E. coli. So here the bacteria The bacteria E. coli is called. So from bacteria the endonuclease is produced. Next question, the specific DNA sequence where echo RI cards is. So the correct answer is option B. So next question, the sticky ends. The sticky ends are produced by the following restriction enzyme except. So the correct answer is option. Which of the following? Option A is the correct answer. Next question, which of the following which of the following restriction enzyme produce blunt ends? So the correct answer will be option D, all of these. Next question, which of the following could be restriction enzyme recognition site? Which of the following could be a restriction enzyme recognition site? So the correct answer option A. Which of the following can give a complementary and palindromic sequence? So the correct answer here is option C. The end of fragments of DNA molecule are sticky due to. The end of fragments of DNA molecules are sticky due to. Option C will be unpaired base, nitrogenous base. So next question, which one of the following palindromic base sequence in DNA can be easily cut at about the middle by some particular restriction enzyme? So the correct answer is option C is the correct option. Next question, which one of the following DNA sequences qualifies to be designated as palindromic sequence? So the correct answer here will be option D. So if you read this sequence from left and from right, the sequence will be same. If I read this sequence from the right sides, it will A G C G C T. If you read this sequence from left sides, it will be A G C G C T. So the same sequence. So the correct is palindromic sequence. So next question, an enzyme catalyzing the removal of nucleotides from the end of DNA is. So the correct answer will be exonuclease. The which of the given statement is correct in the context of observing DNA separated by agar gel electrophoresis? DNA can be seen in visible light. DNA can be seen without staining in visible light. Ethidium bromide stained DNA can be seen in visible light. Ethidium bromide stained DNA can be seen under exposure to UV light. So the correct answer will be option D. So the correct answer is option D. Next question, in agarose gel electrophoresis, DNA molecules are separated on the basis of size only, charge to size ratio, charge only, all of these. So the correct answer is option A, size only. Next question, find out the wrong statement. So udder cell, option A, udder cell, a somatic cell, is used to produce cloned sheep by nuclear transplantation method. Option B, in pedigree analysis, a person immediately affected by an action is called propositus. Mobile genetic element transposons were visualized by Barbara McClinton. Dr. Ian Wilmot produced a clone sheep called Dolly DNA. And option E, DNA ligase are used to cleave a DNA molecule. So the The wrong statement is here. Wrong statement is here option E, because DNA ligase is used to con used to it is not used to cleave the DNA. It's join it used to join two DNA fragment. So next question, the role of DNA ligase in construction of recombinant DNA molecule, formation of the phosphodiester bond between two DNA fragments, formation of hydrogen bonds between sticky ends of DNA fragments, next ligation of all purine and pyrimidine bases, and option D, none of the above. So the correct answer is here option A. The formation of phosphodiester bond between two DNA fragments will be the correct answer for this question. Next question, DNA ligase was discovered by, so DNA ligase is discovered by option C will be the correct answer, Gilbert. Next question, the linking of antibiotic resistance gene with the plasmid vector become possible with DNA ligase will be the correct answer here. So I did not read the all options from this. The enzyme used to join the DNA fragments, again the answer will be DNA ligase. From DNA ligase, various type of question have been asked in different type of exams. So next question, construction of a recombinant DNA involves, so option A, cleaving and rejoining DNA fragments with endonuclease alone. No, this option is not correct. Cleaving DNA fragments with endonuclease and rejoining them with ligase. So it is the, and let's read the next option also, cleaving DNA fragments with ligase. So this option is also not correct. Cleaving and rejoining DNA with ligase alone. So this option also not correct. So the correct answer is here option B. So in recombinant DNA technology or recombinant construction of a recombinant DNA, mainly it involves two steps. First, the DNA fragments it cuts by restriction endonuclease, and then a gene is inserted with this DNA and rejoined by the DNA ligase. So the correct answer for that reason, the correct answer is here the ligase cleaving the DNA fragments with endonuclease and rejoining them with ligase. Next question, question number 49. Enzymes that cleaves nucleic acid within the polynucleotide chain is known as endonuclease, aryl sulfatase, endonuclease, and phosphotriesterase. So here the correct answer is option A, endonuclease will be the correct answer here. Okay, let's go to the next question here. The first plasmid used successfully to clone a vertebrate gene by Cohen and Boyer. So the correct answer is here option C will be the correct answer. Next question, R plasmid are important because they control bacterial reproduction. Option B, control bacteria against mutation. Option C, make bacteria resistant to antibiotics, and option D, can be used to produce transgenic animals. So the correct answer is here option C, make bacteria resistant to antibiotics. Next question, plasmids that carry genes to provide resistance to antibiotics are called R plasmids, RA plasmids, C plasmids, and TI plasmids. So the correct answer will be here option A, R plasmid. Next question, which of the following is not correct statement about the plasmid? Option A, It is extra chromosomal DNA in bacteria. It is not an integral part but insert genetic material. Option C. Host chromosomes can be integrated with the plasmids, and transfer of the plasmid can be done from cell to cell without killing the host. So the correct answer is here is option B. It is not an integral part but insert genetic material. The plasmid occurs in option A bacteria, option C chloroplast, option C virus, and option D chromosome. So the plasmid is always occurs in bacteria. Next question: What are true? What are true of plasmids? They are found in virus. No. They are remain parts. They are main parts of the chromosome. No. They are widely used in gene transfer. They contain gene for vital activities. So the correct answer is where the option C. They are widely used in gene transfer. Next question: Plasmids. Again from plasmid. Plasmids are outgrowth of mitochondria. No. Outgrowth of cell membrane. No. Outgrowth of nuclear membrane. No. It's a extra chromosomal circular genetic material. So next question again from plasmid. Plasmid is a double stranded circular DNA, extra chromosomal linear DNA, single stranded DNA, and none of the above. So the correct answer is here option A, double stranded circular DNA, and it is found available in bacterial cells. And it is next question: Plasmids are ideal vectors for gene cloning as they can be multiplied by culturing, replicate freely outside the bacterial cells, are self-replicating within bacterial cells, and can be multiplied in laboratory using enzymes. So the correct answer is option C, are self-replicating within the bacterial cells. So the next question is: Plasmids are extracellular circular DNA molecules which have their own point of replication and can replicate independently. Option B, which have their own point of replicant but cannot replicate independently. Which do not have their own point of replicates and cannot replicate independent of bacterial chromosome DNA. None of the above. So the correct answer is option A. Plasmids have their own point of replication and can replicate independently. So the correct answer option A will be the correct answer. So next question, let's go to the next question here. Okay, let's begin. The plasmids are suitable vectors for gene cloning because these are small circular DNA molecules which can integrate with host chromosomal DNA. Option B, these are small circular DNA molecules with their own replication origin site. And option C, these can settle between prokaryotic and eukaryotic cells, and these often carry antibiotic resistant genes. So the correct answer will be option B. Option B will be the correct answer. One of the key factor which makes the plasmid the vector in genetic engineering is that it resistance to antibiotics. One of the key factor which makes plasmid the vector in genetic engineering is that option A, it is resistance to antibiotics. It is its ability to carry a foreign gene. It resistant to restriction enzymes. Its ability to cause infection within the host. So the correct answer will be option B. It its ability to carry a foreign. So plasmids are option C DNA, viral DNA, mitochondrial DNA, and circular extra chromosomal DNA in bacteria. So the correct answer is option D. Next question. As apart from DNA in bacterial nucleoid, there is a circular extra chromosomal DNA called plasmid. Will be the correct answer for this question. Okay, let's go to the next question here. The vector tDNA is a Thermus aquaticus, Salmonella typhimurium, Agrobacterium tumefaciens. So the correct answer will be option C. Next question. In recombinant DNA technique, the term vector refers to the plasmid that transfer foreign DNA into a living cells. No, the cosmids that can cut DNA at a specific base sequence, plasmids that can join different DNA fragments, cosmids that can degrade harmful protein. So the correct answer is option plasmids that can transfer foreign DNA into a living cells. So, next question. Which of the following are used in gene cloning? Nucleoids, mesosomes, lomasomes, and plasmids. So, the correct answer is option D, plasmids. Next question. Which one of the following is used for vector for cloning the genes into higher organism? So the correct answer is option A, retrovirus. Which of the following is used as vector for cloning genes into higher organisms? So the retrovirus will be the correct answer. Next question. What is the function of a vector? To help to amplify the DNA. Option B, allows cells to take up foreign DNA, destroy cells that do not contain cloned DNA. Option D, carries cloned DNA, enabling it to replicate in the clone cells. So the correct answer is option D. Option D will be the correct answer. An ideal vector must possess multiple cloning sites, an origin of the replication, and a gene encoding a restriction enzyme, a gene encoding resistance to an antibiotic. So the correct answer will be option N, origin of replication. During gene cloning, which is called gene taxi, vaccine, bacterium, plasmid, and protozoa. So the correct answer is option B will be the correct answer, plasmid. A plasmid that have been cleaved with EcoR can hybridize with another plasmid that has been digested with EcoRI. Option B, in southern blot and digested with hint 3 and cleaved with BAM-H. So the correct answer will be option A, the digested with EcoRI. Which of these are correct in view of the genetic engineering? It uses resistant plasmid PBR322. Option B, DNA molecules are broken down by topoisomerase. Option C, recombined DNA or rDNA is called chimeric DNA, and both. Option D, both A and C. So the correct answer will be option D, both A and C. So next, going to the next question, I'll take little Okay, let's begin. Next question. The construction of the first recombinant DNA was done by using native plasmid of E. coli, B. thuringiensis, Salmonella typhimurium, D. yeast. So the correct answer will be here option B. Salmonella typhimurium. So the next question, plasmid BR PBR 322 used in biotechnology is yeast parasites, M32 phage and cloning vehicles. So the correct answer will be cloning vector. Next question, identify the plasmid. So the plasmid is B30 BR322. So let's move to the next question. Which of the following is a recent application of the genetic engineering in diagnostic technique? So here four options are given. You have to choose the correct answer. So the correct answer will be the ELISA test. So next question. Let's move to the next question. An antibiotic resistance gene in a vector usually helps in selection of competent cells, recombinant cells, transformed cell, and none of these. So the correct answer is option B, transformed cells. So let's go to the next question. The most important feature in a plasmid to be used as a vector: its size, origin of replication, the presence of a selectable marker, the presence of sites for restriction endonuclease. So the correct answer is option B, origin of replication site. Next question, the crown gall disease is in plants is caused by TI plasmids. So next question, a tumor inducing plasmid widely used in the production of transgenic plant is that of Escherichia coli, Bacillus thuringiensis, Staphylococcus aureus, and Agrobacterium tumefaciens will be the correct answer for this question. Next question, TI plasmid used in genetic engineering is obtained from Agrobacterium rhizogenes, Agrobacterium tumefaciens, Bacillus thuringiensis, Pseudomonas syringae, and Bacillus subtilis. So the correct answer will be the option B, Agrobacterium tumefaciens. The tumor inducing capacity of Agrobacterium tumefaciens is located in large extrachromosomal plasmids called. So the correct answer will be option C, the TI plasmid. Next question, which one of the following is commonly used in transfer of foreign DNA into crop plants? So the Agrobacterium tumefaciens will be the correct answer for this question. Next question, polyethylene glycol method is used for energy production from sewage, gene transfer without a vector, seedless fruit production, and biodiesel. So the correct answer will be the correct answer. Next question, insect tolerant gene Bacillus thuringiensis is introduced using TI plasmid of Escherichia coli, Arabidopsis thaliana, Haemophilus influenzae, Agrobacterium tumefaciens. So the Agrobacterium tumefaciens will be the correct answer for this question. So next, let's move to the next question here. The stable enzyme tag and PFU is isolated from thermophilic bacteria are DNA ligases, DNA polymerases, RNA polymerases, and restriction endonuclease. So the correct answer is option C. So option C is the DNA tag and PFU is the DNA polymerase. Next question: gene amplification using primer can be done by gene gun, electrophoresis, microinjection, polymerase chain reaction will be the correct answer. Next question: which of the following steps are catalyzed by Taq polymerase in a PCR reaction? Extension of primer end on the platelet DNA, annealing of primers to template DNA, denaturation of the DNA template DNA, all of the above. So the correct answer is option A, extension of primer end on the template DNA. So pause the video and read the question and try to answer this question. So next question: PCR proceeds in 3 distinct steps governed by temperature. They are in order of. So denaturation, annealing, and synthesis. So the option D will be the correct answer for this question. Next question: the source of Taq polymerase used in PCR is called thermophilic fungus, mesophilic fungus, thermophilic bacterium, halophilic bacterium. So the correct answer will be the thermophilic bacterium will be the correct answer. Next question is polymerase chain reaction generates a fragment of a distinct size even when an intact chromosome is used as template. What determines the boundaries of the amplified fragments? So here 4 options are given and you have to choose the correct answer for this question. So now the option A will be the correct answer. The sites to which the primer anneal. So next question: the polymerase chain reaction use Taq polymerase because rather than a DNA polymerase from E. coli, because Taq polymerase is a heat stable, easier to obtain, can denature a double-stranded DNA template, can initiate DNA synthesis at a wider variety of sequence. So the correct answer is option A, heat stable. Which of the following enzyme is used in polymerase chain reaction? So Taq polymerase is used in polymerase chain reaction. So next question, let's move to the next question here. Let's move to the what is utility of a bacterium Thermus aquaticus? It is used in RFLP mapping. No. It is used to create recombinant plasmids. No. It is used in automated DNA sequences. It facilitates the polymerase chain reaction. So the correct answer is option D. Next question is the paleontologist unearthed a human skull during excavation. A small fragments of the scalp tissue was still attached to it. Only little DNA can be extracted from it. If the gene of the ancient man need to be analyzed, the best way of getting sufficient amount of DNA from this extracts is option A, hybridizing the DNA with a DNA probe. No. The option B, subjecting the DNA to gel electrophoresis. No. Subjecting the DNA to polymerase chain reaction will be the correct answer, and treating the DNA with restriction endonuclease is not correct. So the correct answer is option C. Let's go to the next question. The polymerase chain reaction is a technique that is used for in vivo replication of the DNA. Option B, in vivo synthesis of the mRNA. Option C, in vitro synthesis of the mRNA, and in vitro replication of the specific DNA sequencing using thermostable DNA polymerase. So So the correct answer will be the option D. In vitro replication of the specific DNA sequence using thermostable DNA polymerase. So next question, the polymerase chain reaction is concerned with DNA amplification, DNA proofreading, DNA repairing, DNA replication. So here it is the question, it is very confusing. So the correct answer will be the option amplification. Who among the following was awarded the Nobel Prize for the development of PCR technique? Carey Mullis, Arthur Kornberg, and Herbert Boyer and Hargobind Khorana. So Carey Mullis the Carey Mullis will be the discoverer or is the discoverer of PCR technique. PCR technique, and he won the Nobel Prize for his discovery. The PCR technique was invented by, again the same question, Carey Mullis will be the Carey Mullis is the next. Thermal cycler is used in the reaction radioactivity chemical reaction. Polymerase chain reaction will be the correct answer, and enzyme catalyzed reaction is not correct for this question. Next question, the manipulation of DNA in genetic engineering become easy due to invention of dot blot, instant blotting, polymerase chain reaction, and enzyme linked immunosorbent assay. So the manipulation of DNA in genetic engineering become easy due to the invention of. So the correct answer is option C will be the correct answer, polymerase chain reaction. Next question, PCR stands for polymerase chain reaction will be the correct answer. Okay, let's move to the next question. Okay, so the first mammalian clone Dolly was created by Ian Wilmot, T.H. Morgan, Robert Briggs, and Thomas King and Gregor Mendel. So for this question, the correct answer will be option A, Ian Wilmot. So recently cloned species is Dolly Goat, Dolly sheep, Molly sheep, and none of these. So here the correct answer is option C, Dolly sheep will be the correct answer. Next question, the following can be described as clones: a colony of bacteria derived from a single cell by asexual reproduction, the mother of the sheep Dolly and Dolly, the identical twins arising out of the single egg, all of the above. So the mother of the sheep Dolly and Dolly. So bacteria. So the correct answer is here option D, all of these. Next question, the clonal cell line can be obtained by tissue system, tissue fractionation, tissue culture, and tissue homogenization. So the correct answer is here option B, tissue culture. From tissue culture it is. Next question, a new method of harvesting stem cells is known as cloning, entrapping, sporogamy, cygogamy. No, this is the cloning will be the correct answer. Genetically similar individual of a species are cloned. So the clone will be the correct answer. Next question, cloning gene is a process where gene is cloned in animal. Fragments of DNA are transferred from one organism to another, usually carried on a DNA vector. Fragments of DNA cloned in the same organism using carrier DNA is cloned in plants. So the cloning, the correct answer is option B. The fragments of the DNA are transferred from one organism to another, usually carried on the DNA vector. So next question, the offspring produced through which of the following processes are not exactly similar to their parents? So sexual reproduction, parthenogenesis, asexual reproduction will be the correct answer for this. The again I will read the question. The offspring produced through which of the following processes are not exactly similar to the parent? So the asexual reproduction, the uh, offspring are exactly similar with their mother. This option is not correct. So in sexual reproduction, the offspring are not exactly similar to the so for this question the correct answer will be the option C. Next question, uh, you have to you have to match the both options. So here are the scientists and their achievements or achievement or discover invention in son. So Arthur and Arver and Smith, so they discover endonuclease. Feldman, so Feldman will be option. Development of transgenic plants with agrobacterium tDNA. So option C, Mullis, he discovered the PCR, and Temin and Baltimore, who discovered reverse transcriptions. Then if you arrange A match with 2, B match with 1, C match with 4, D match with 3. So the correct answer is here option A. Okay, let's move to the next question here. The organism which is used for gene transfer in higher organism. So option A, E. coli, option E, Acetobacter. So and another option is Bacillus thuringiensis and Agrobacterium tumefaciens. So the correct answer will be option D, Agrobacterium tumefaciens. So let's move to the next question. Widely used tool in genetic engineering of crop plants involves microinjection, protoplast fusion, transposons mediation, and agrobacteria mediation. So the correct answer is here option D. The next question. Let's move to the next question here. When a cow is crossed with a bull and female progeny yielding more yield than its mother, from this it is inferred more number of genes for high yielding milk are inherited only from the female parent. More number of genes for high yielding milks are inherited only in the male parent. More number of genes for high yielding milk are inherited from both parents. The progeny through mutation achieved more number of genes for high yielding meal. So from these options, you have to choose the correct answer. So now the correct answer for this question is option C will be the correct answer. More number of genes for high yielding meal are inherited from both parents. So this is the correct answer for this question. Next question, significance. Next question, the significance of heat shock method in bacterial transformation is to facilitate the binding of DNA to the cell wall, expression of the antibiotic resistance gene, uptake of DNA through membrane transport proteins, uptake of DNA through transient proteins in the bacterial cells wall. So the correct answer is option D, uptake of DNA through transient pores in bacterial cells wall. So the next question, the following steps are used to make a recombinant cell. What is the fourth step? So the fourth step is here is option D, clone cell containing vector. The first simian cloning was carried out by. So the correct answer is option B, Ron Wolf. Next question, Andy is the clone cat, man, monkey, bull, sheep. So The correct answer is here option E, monkey. Next question. The choose the correct statement with the reference to the dolly. She was created. So she was created by, you read the options, and here I am telling you the correct answer will be option C. And pause the video and read the all options, and you also next question. Next question here. Okay, today that's for all.